Good afternoon. I am Matti Hämäläinen from Massachusetts General Hospital and Harvard Medical School, and today my topic is real-time signal processing and source localization. My contents is the following. First, I will give some motivation why we actually need real-time software. Then I will show 3 examples of devices in our laboratory which we have built software for. And then I will discuss in more detail our real-time software, which is called MNE-CE. Present electrophysiological acquisition tools require to be compatible with multiple platforms and devices for data acquisition, like MEG, EEG, ECOG, and stereo EEG. However, practically this hasn't been possible up to now, and as a result there are several different pieces of software which have to be used depending on the device. These acquisition software packages typically contain only few real-time features, maybe real-time averaging, but more sophisticated features are rare. Furthermore, integration of results collected with different techniques is difficult and slow. This stems really from the fact that the software packages are different for different devices and then don't work well together. And most real-time packages are limited to sensor-level analysis. That is, we cannot estimate the sources in the brain and look at the time courses, for example. On the other hand, there are many offline-only packages for source analysis, and they can be used to understand better what the non-invasive and invasive electrophysiological data mean. Additional motivations for developing real-time analysis methods are, first of all, we can provide a lot of quality assurance during the measurement by being able to analyze the data to at least to some extent already during the recording. And then if we think about experiments, we can fine-tune the experiments during piloting by looking at the results of the acquisition and then changing the details of the experiment if necessary, if we find something that would be better optimized by changing the experiment. And of course, if we can do some of the analysis in real time, we speed up the cycle from acquisition to publication or clinical diagnosis considerably. And as I already mentioned, device independence means that we can not only accommodate different devices, but also if we build new devices, we can prototype them faster because we can use the same acquisition software for all of them. Furthermore, brain stimulation and brain recording devices combined devices are becoming more common nowadays, and we can imagine with real-time software a closed-loop operation where we record, stimulate the brain, and record the results of the stimulation, and actually may change the stimulation according to the results of the recordings. And I would say in general that with proper real-time software, the experiments take the center stage, so that it's really the experiments that are the main point of science, and therefore everything gets more focused on the experiments when we can do also part at least of the analysis in real time. We have built recently 3 different devices, each of which requires specialized real-time software which is not available otherwise. First of them is the so-called baby MEG, which is the whole head MEG device for infants at Boston Children's Hospital. This has a dense array of squid sensors close to the baby's head, and also 35 triplets of compensation sensors to disturbances And as you can see, babies are quite happy being recorded in these devices, and it's not scary at all, at least after some accommodation. The second device we have built is what we call a conformal MEG system. This device uses optically pumped magnetometers to record MEG signals around the head. And since the sensors are like the a little bit big EG electrodes, they can be positioned at will around the head, and they accommodate different head sizes. And here is the practical device being assembled in our laboratory, and on the right we have the acquisition electronics and electronics software, which has to be then connected to higher level software which we are building to actually acquire and store data from this device. The third device we have built is a combined stimulation and acquisition system. It combines transcranial magnetic stimulation and MEG, and it has 16 liquid nitrogen cooled TMS probes, which can be used to stimulate different parts of the brain selectively. And then it has 25 optically pumped magnetometers for recording the MEG. The arrangement is schematically shown here on the lower right, but this is the actual device in the laboratory after it was assembled together. And on the right we have a subject sitting in a chair under this helmet, which has the TMS coils, 16 of them, and MEG recording sensors. Our software is built under a project which is called MNE-CE, where CE stands for Clinical Electrophysiology. This is a UO1 supported by NIBIP, and its title is Device Independent Acquisition and Real-Time Spatiotemporal Analysis for Clinical Electrophysiology Data. So this software, as the name of the grant implies, tries to fulfill many of the requirements I have mentioned before. This software has 3 modes of operation: a true real-time operation mode, then a playback mode in which previously recorded data is played back from a file so that we can test how well the real-time operations work without actually having to go to the device for this testing and software development purposes. And finally, there's an offline mode, because we have assembled some components of the software to make offline tools which we can then use for subsequent more detailed analysis. And the benefits of this software package include that its device dependence is well isolated in the small portion of the code. There's already existing solid infrastructure for real-time source estimation. It's fast, it's C++ code compiled, it supports multiple operating systems, and we have an established development workflow on the right, so that since this software is professionally built, we can eventually ask for an FDA approval for the software. And finally, because of the way the software is constructed, it can be used clinically and eventually also commercialized. In real-time source estimation, we use structural MRI, shown on the top, from which we built the geometry model of the head for source estimation, and also find where the cortical surface is so that we can have the source locations fixed. And on the basis of this information and the head position, which comes from the data containing signals from the so-called head position indicator coils, we built a K-matrix and used this K-matrix together with the data and the noise statistics derived from the data to estimate the sources in the brain. And here's an estimate of a pipeline using the MNE-CE software, which works in real time, connects to an MEG system. Then we have a signal space projection and a filtering module which suppresses noise. We calculate noise covariance. In the next step, average the data, and below is shown the one source estimate which is called regionally clustered minimum norm estimate. So for the TMS MEG device, we have a more complicated situation. We first of all need to decide where to stimulate, and for this decision we need the forward solution for TMS, and then we find the best currents to feed to the coils, to the 16 coils, to stimulate a certain region of the brain. Then in the middle we have the stimulation and recording using the stimulation parameters which we decided we stimulate using the 16 coils, and then we record MEG and or EEG from the brain and decide where the brain is activated as a result of the stimulation. And on the basis of this result, we can then decide new targets or just look at the excited pattern. In the future, we envision that these kinds of real-time software packages can also change the intervention in real time. For example, we can decide where to place the invasive electrodes by measuring and analyzing the data in real time from them, and especially we can analyze data in real time during surgery, and we can decide to stop long-term monitoring of epilepsy patients if we have seen that enough data has been collected for sufficient diagnosis. And also, since the software works equally well with different devices, we can also readily compare invasive data and previously collected non-invasive data to understand how they relate to each other. In summary, many experimental devices need solid acquisition and real-time analysis software. This is actually not limited to MEG and EEG devices, but you can imagine that the same is true for other imaging devices as well. MNEC is a software package which provides a platform for creating both real-time analysis tools and parallel methods for offline analysis. Since we have compiled C++ code and established development process, we can eventually think about FDA approval of the MNEC and therefore pave the way for clinical use of the software. And the many components of MNEC can also be used to create tools for basic research applications. They can be either real-time or offline analysis applications, but they can use these basic tools we have already built. Thank you.